If we start at the sunlit surface of the ocean and drop straight down, the environment changes rapidly. Over 70% of Earth's oceans exist in a state of permanent, crushing darkness. Below 200 meters, conditions degrade rapidly. In the bathopelagic zone, temperatures hover near freezing, and hydrostatic pressure is immense. Meaningful solar radiation ceases completely. To survive, organisms adapted biological alchemy, converting chemical energy into visible light to camouflage, communicate, hunt, and evade. Some microscopic dinoflagellates use this light as a literal burglar alarm. When a predator attacks, the dinoflagellate emits a sudden blinding flash. This illuminates the attacker in the dark water, drawing in an even larger secondary predator to consume the threat, allowing the original prey to escape. Bioluminescence has evolved independently dozens of times across the tree of life. Down here, producing visible light is an absolute evolutionary imperative for survival. When humans want to illuminate a space, we rely on massive electrical grids and physical resistance, generating light largely as a byproduct of intense heat. This chart compares biological light generation to standard artificial lighting. Marine bioluminescence converts chemical energy into photons with greater than 95% efficiency, producing almost zero thermal waste. This cold light obeys the same strict laws of thermodynamics that govern our power grids. It simply executes those laws with a precision that human engineering struggles to replicate. By studying how these organisms generate light without losing massive amounts of energy to heat, researchers are reverse engineering a zero-emission blueprint for future technologies. To understand exactly how they achieve this, we have to look past the scale of the ocean trenches and zoom directly into the cytoplasm of a single marine bacterium. The biochemical paradigm relies on two main components. A light-emitting small molecule, called a luciferin, docks with a catalyzing enzyme called a luciferase. When oxygen is introduced, the resulting chemical intermediate becomes highly strained. As it relaxes back to its ground state, it releases a photon. In marine environments, this emission is almost always blue, because blue wavelengths travel the furthest through the dense medium of seawater. But producing these photons is a massive drain on cellular energy. To avoid burning reserves when floating alone in the vast ocean, bacteria utilize a complex communication mechanism called quorum sensing. The bacteria constantly secrete chemical signaling molecules into the water. If the population density is low, the signals drift away. But when they are packed tightly together inside the light organ of a host squid, the local concentration of these molecules crosses a critical threshold, triggering the entire colony to simultaneously ignite. They conserve their energy at the cellular level by relying on perfectly timed molecular communication. In the summer of 1961, the attempt to isolate these mechanisms brought researchers out of the deep ocean and up to the coastal waters of Friday Harbor in the Pacific Northwest. Chemist Osamu Shimomura and his team collected tens of thousands of crystal jellyfish from the Puget Sound, extracting their luminous tissue in an attempt to isolate the exact chemical pathway. After months of failing to trigger the glow with standard cellular energy, Shimomura poured a discarded sample into a lab sink that contained overflow seawater. The liquid instantly flashed with a brilliant blue light. The reaction was driven entirely by trace calcium ions. But the live jellyfish floating in the ocean glowed green. By studying the extracts further, Shimomura found a secondary protein. It absorbed the initial blue emission and re-emitted it at a lower energy as visible green light, a structure now known as green fluorescent protein, or GFP. Because its glowing fluorophore is entirely self-contained within its protein structure, scientists learned to splice the GFP gene into non-luminous cells. It provided a molecular flashlight, allowing researchers to track the real-time movements of proteins and map genetic expressions inside living hosts. Today, the applications of this marine chemistry have scaled up from basic cellular tracking directly into the firing neural networks of the human brain. Traditional deep brain stimulation, used to manage severe neurodegenerative conditions like Parkinson's disease, requires surgically implanting metal electrodes deep into the tissue to deliver electrical shocks powered by external batteries. Researchers are now testing a genetic alternative called bioluminescence resonance energy transfer, or BRET. Instead of physical fiber optic cables, Brett uses the body's own internal biological light to trigger specific neurons. Scientists genetically tether a luciferase enzyme directly to a light-sensitive ion channel on a target neuron. When a specific chemical substrate is administered to the subject, the tethered enzyme oxidizes it and flashes blue, instantly opening the channel and forcing the neuron to fire. The process turns the subject's internal biochemistry into a highly specific, non-invasive tool for neurological repair. Moving outward from the human body, this same structural logic is now being tested in our agriculture and physical infrastructure. By mapping the specific enzymatic pathways of luminous fungi, synthetic biologists have successfully spliced self-sustaining light directly into the genomes of common plants. Because the plant continually regenerates the necessary chemical inputs through its own natural metabolism, the glow operates on a perpetual autonomous loop. Urban planners are researching how to apply this to our cities through algae bio-reactive building envelopes. By integrating genetically optimized glowing microalgae into glass facades, cities could eventually sequester massive amounts of carbon dioxide while providing soft ambient street illumination. These biological systems will not immediately replace our global electrical grid. What they offer is a functioning, infinitely renewable standard built by millions of years of evolutionary pressure. By decoding the chemical architecture of the deep ocean, we are learning how to light our world using the chemical energy we already possess.